So good morning to everyone. First, I would like to thank to Gordon for organizing again the Human Glycome Project meeting. So this time with the opportunity of bringing the Glycan Switch Consortium, which is great. So today I'm going to talk about some new insights that we got from the EU-funded Glycan Trigger project, which is pretty much related on how glycans and how glycome can be important as triggers of the health to inflammation transition. And then more at the end of my presentation, I will show you how the two projects that we are involved in, the glycan switch and glycan trigger, can be somehow interconnected through the way that the glycome can be really a common denominator of many immune-mediated diseases, such as the case of IBD for glycan trigger and RA for glycan switch. So we all know the relevance of glycans in health and life sciences. And my group at the University of Porto, in the Institute for Research and Innovation in Health, we are pretty much interested in this dynamic role played by glycans as powerful immune regulatory molecules. So, and we are interested on how, from one side, glycans are important in establishing powerful immune suppressive anti-allergenic pathways that we know to occur associated with cancer progression, but also how other types of glycans are also important in triggering pro-inflammatory and autoreactive responses that we know to occur in chronic inflammation and autoimmunity, which is the focus of my talk of today. And the relevance of glycans in immunity comes from the evolution. So glycans are actually very important evolutionary molecules relevant for self and non-self discrimination. And this comes from the seminal paper mentioning that nothing in glycobiology makes sense except in the light of the evolution, in the way that we mammalians are equipped with very complex glycan structures. We are equipped with the capacity to synthesize tetrantinary glycans, silylated glycans, fucosylated glycans. However, the microorganisms, they synthesize much less complex glycan structures and mainly mannose and rigid glycans. But what happens as a consequence of the evolution is the sharing of some glycan structures between us and the microorganisms that give rise to the concept of glycan mimicry. And this similarity, this overlap of glycan structures between us and the microorganisms, giving rise to the concept of the glycan mimicry, means that, for example, the microorganisms, they can engage in glycan mimicry as a way of decorating themselves with glycan structures similar to the host, and in this way infecting the host. But what happens is also the incapacity of the immune system within this glycan mimicry process to distinguish between self-glycans and non-self-glycans, and these may end up in some autoreactive responses as these glycan-driven immunological pathway that we described to occur, for example, in lupus. But the relevance of glycans in immunity can actually be scaled up by the fact that our immune system are equipped with glycan sensing properties, because all of our immune cells, the B cells, T cells, and myeloid cells, express such a variety of glycan binding receptors, the so-called cytoplactins, galactins, and Siglecs, establishing powerful glycan-derived immune regulatory networks or hubs that are very important to define the nature and the fate of the immune response. So when perturbed, this glycan and glycan-binding protein interaction, we may end up in autoreactive responses, such as the case that occur in IBD or in RA. So starting to tell you about the relevance of glycans as master triggers of health to inflammation transition, today I'm bringing two examples. So one is what happens in terms of the IBD, and then what happens also as a way of glycans being a denominator of immune-mediated diseases, what happens, for example, in RA. So starting in the mucosa glycome. So this actually is the right place to ask our questions about the relevance of glycans in immune response, because the mucosa glycocalyx couldn't be more rich in terms of abundance and diversity of glycan structures encoding powerful immune regulatory properties. So talking about IBD as the disease model, so IBD is a chronic inflammatory disorder of the gastrointestinal tract. It comprises essentially Crohn's disease and ulcerative colitis. And both diseases, they learn from these same mechanisms of a huge infiltration of inflammatory cells that essentially destroys the glandular architecture of the intestine. But what happens is that the patients frequently alternate between periods of activity of the disease with periods of remission of a controlled disease that, unfortunately, in an unpredictable fashion, may end up again in periods of exacerbation of the disease, the so-called flares. And the current clinical unmet needs in the clinical management of these patients is the fact that we do not have currently biomarkers or molecular tools able to predict the severity of these flares. We do not have biomarkers able to predict the disease score, so who is likely to develop an aggressive or complicated disease, and we do not have biomarkers to predict the response to therapy. But nowadays, I should say that the question that is the closest to our hearts in the group, that would be quite transformative to the patients and to the families, is how to predict IBD development. Because if we're able to predict, we'll be able to prevent. So IBD, as many other inflammatory diseases and autoimmune diseases, is a multifactorial disease. It's a result of contribution of four main pillars, I would say: environmental trials, a dysregulation of the immune response, the contribution of the microbiome, and of course genetic susceptibility of the host. And we can do, being a glycobiologist aficionada, we can put glycans in the middle and in the center as a truly layer of information that can actually articulate with all of these factors. How? So we know that glycans are important in the modulation of the microbiome composition, and this is something that we are actively exploiting in the group. We know that there are some genetic polymorphisms in glycogenes associated with immune-mediated diseases. We know that glycans exert powerful immune regulatory functions, and we can find glycans in nature, such as in diet. So our accumulating evidences over the past, I would say, decade or so, led us to the fact that we gather a compelling body of evidences showing that the glycome and the changes in cellular glycosylation are important in the immunopathogenesis of IBD. So starting some years ago, we started to demonstrate that in active ulcerative colitis patients, they exhibit a deficiency on branched complex and glycans synthesized by the GNT5 enzyme and encoded by the MGAT5 glycogen. And this deficiency on the branched glycans was observed specifically at the level of the mucosal T cells. And this deficiency in mucosal T cells glycosylation was due to a downregulation of MGAT5 glycogen that led us to propose this mechanism, saying that in an hyperimmune response, in an uncontrolled immune response, mucosal T cells exhibit a deficiency in glycan structures, contributing to an increase in the T cell signaling. On the contrary, in a normal or controlled immune response, mucosal T cells are glycosylated with branched glycans, and this controls to T cell mediated immune control. So we decided to test whether the glycosylation reprogramming of the gut mucosa could be a way of achieving immune control. And we tested whether the metabolic supplementation with glycans can be actually a source of powerful immune regulatory molecules to control the immune response. So Anadias was a talented PhD student at that time in the group that carried out this demanding study, because this was essentially dependent on human samples. So what we did was to isolate CD3 positive T lymphocytes from the fresh colonic biopsies of the patients, and we started to metabolically supplement with increasing doses of glycans. And we managed to repair that deficiency on the branched glycans on T cells. But the good thing is that we observed that this enhancement on the branching glycosylation of these T cells from the patients was associated with a suppression on T cell proliferation, of an inhibition in the secretion of these potent pro-inflammatory cytokines, and a control in the T cell receptor signaling. We validated this in mice models as a preclinical assay. We used the MGAT5 knockout mice that essentially mimics the deficiency on the branched glycans that we observed in patients. We treated the mice with metabolic supplementation with glycans, and we were able to observe the suppression of disease progression and severity, essentially because we are able to control the Th1-like immune response. So these altogether allow us to propose that glycans exert powerful immune regulatory properties, controlling the T-cell mediated immune response, setting the stage to an investigator-initiated clinical trial that we are now carrying out through the Glycan Trigger project. But besides the fact that glycans are important, or cellular glycosylation alterations are important in the immunopathogenesis of the disease, we also gather evidences, and this was a project that we did in collaboration with AbbVie, that glycans, they can be actually important biomarkers with prognostic performance. And we demonstrated that assessing the levels of glycosylation at the tissue level, so at diagnosis of ulcerative colitis patients, this is able to predict, for example, the response to therapy. So all in all, we gathered the evidence that glycome, or the changes in glycome, are important in the immunopathogenesis of IBD by demonstrating that patients exhibit a deficiency on cellular glycosylation at the level of the T cells, that if we metabolically glyco-reprogram the gut mucosa, we can achieve immune control, setting the stage for a clinical trial. We also demonstrated the relevance of glycans as powerful biomarkers associated with the response to therapy. We also demonstrated that there are some genetic polymorphisms or variants in MGAT5 glycogen that can explain these changes in the cellular glycosylation. And we are also exploiting the role of the changes in glycosylation in the microbiome composition and dysfunction, and how this is associated with the onset of IBD. And this is a recent review that we wrote in Semel in Immunology. So, but as I told you in the beginning, the question that is of major interest for us is how to predict IBD development. So in fact, in IBD, predominantly in Crohn's disease, like in many other immune-mediated diseases, which is the example of RA, there are mounting evidence of the existence of a preclinical phase that can last years before the diagnosis, where immunological changes can occur many years before the clinical manifestation or the clinical symptoms of the disease. And this is actually where we are and we are interested in. So the question is, what is the role of the mucosa glycome, or what is the role of the glycome in this health to inflammation transition? And to study the preclinical phase, we needed to study serum, and I will tell you how we did this. This was actually a collaboration with Mount Sinai in New York and with the US Department of Defense, in which we had access to this beautiful military population with access to thousands of samples representative from the preclinical phase, so up to 6 years before the diagnosis, in which we analyzed the serum glycome composition. So talking about the serum glycome, we couldn't avoid to talk about IgG glycosylation. So this morning we were already talked about IgG, and Gordon introduced that. So we all know that IgG glycosylation is very important in defining the effector functions of IgG, at least for the FC domain. We had this seminal publication in the 80s in RA, in which changes in IgG glycosylation was observed before disease onset. And we also have evidences for the existence of dozens of different glycan variations that can be detected in the healthy individual. So we asked the question on whether IgG glycome heterogeneity among the healthy population could translate into a better understanding of the preclinical phase of IBD. So we managed to analyze this in collaboration with Gordon and at Ginos. We analyzed 3,000 samples from the military populations in the preclinical phase, so 6 years before, 4 years before, 2 years before, and at diagnosis, from individuals that later developed Crohn's disease, later developed UC, and those that remained healthy. So Joanna and Claudia are respectively a postdoc and a PhD student in the group that carried out this project, and we started to observe, actually in the very first batch of analysis, a significant change in the IgG glycome profile that remained stable over time and were correlated with the Crohn's disease development, but importantly also with the risk for developing complicated disease years before the diagnosis of the disease. So then we established a correlation analysis between the IgG glycome profile and other antimicrobial antibodies, such as the case of ASCAM. So ASCA is anti-Saccharomyces cerevisiae antibodies. It was discovered by one of the collaborators and partners of the Glycome Trigger project, Jean-Frédéric Colombel, that discovered ASCA many, many years ago. It's an antimicrobial antibody, but also we correlated with others, such as anti-flagellin antibodies, also detected to be associated with IBD. And we found that the top markers of IgG glycome that we found to be correlated with Crohn's disease development are in the same cluster as antimicrobial antibodies. And in a network analysis to depict correlations between the different IgG glycan traits that we observed in the preclinical phase with the antimicrobial antibodies, we identified this unique linkage, this unique connection between this single glycan trait, IgG2, H3, and 4F1, and ASCA, exclusively with ASCA, pinpointing this glycome ASCA hub that can be detected in the furthest time point from diagnosis. And this was validated also in this way, meaning that only ASCA-positive patients exhibit increased level of this ASCA IgG glycan trait. So at that time, while we were discussing this with the collaborators from Mount Sinai and the US Department of Defense, we raised the question about the relevance of this glycome ASCA hub detected in the preclinical phase. So ASCA, for many years, was taken by the gastroenterologist as a bystander biomarker that was just there with no role in terms of the disease development. But then we asked the question on whether glycome ASCA hub, this ASCA could be really pathogenic in terms of contributing to the disease pathogenesis years before the diagnosis. So, and we decided to prove that in this way. So we isolated ASCA from this military cohort, this ASCA IgG glycoform, and we did the co-culture with dendritic cells and NK cells. So why dendritic cells and NK cells? Because are the ones that exhibit Fc gamma receptor, and we know that IgG Fc glycosylation modulate the effector functions of these cells. So what we started to observe was actually in the preclinical phase, so before diagnosis, this ASCA IgG glycoform was able to activate dendritic cells to lead to the upregulation of the Fc gamma receptor. And because when we block the interaction between ASCA IgG Fc and the Fc gamma receptor from dendritic cells with inhibitory antibodies, we no longer observe the activation of dendritic cells, proving this Fc gamma receptor interaction. And we also observed that this ASCA IgG glycoform is able to reprogram dendritic cells towards a pro-inflammatory activity, as you can see in terms of increase in IL-1 beta, IL-6, IL-8, and TNF-alpha. And then we decided to validate this in an unbiased approach. And to do that, we did RNA sequencing of these co-cultures between the ASCA IgG glycoform isolated from the preclinical phase with dendritic cells. And it was very interesting to observe that at the furthest time point from diagnosis, so 6 years before, there is a clear reprogramming of the dendritic cells in terms of a clear differential gene expression profile, where dendritic cells, they start to exhibit increased expression levels of genes related, for example, with TNF-alpha, NF-kappa beta, and sitaplactin receptors, which is great for us because this equipped dendritic cells with glycan sensing properties. So we also validated this at the functional level. We demonstrated that, in fact, this co-culture between ASCA IgGs and dendritic cells are able to lead to the upregulation, for example, of DC sign and Dectin-2, again, equipping dendritic cells with glycan sensing properties. Together with the increase of the NF-kappa beta signaling that converge into the inflammasome activation and CARD9 years before the diagnosis. And we know that CARD9 has been widely described to be associated with Crohn's disease development. We also did this approach in NK cells. For the sake of time, I will not have time to tell you all of the story about NK cells, but just to let you know that we also demonstrate that NK cells are activated with increase in terms of degranulation and granzyme B expression. But then we needed to have a more complete model to prove the pathogenic role of ASCA. And after many meetings in terms of brainstorming with all of these amazing girls that I have in the group, Joana and Inês. Inês is here in the audience, 2 postdocs in the group, and Claudia and Eduardo, 2 PhD students. We end up in this animal model. So we decided to start immunizing mice with the antigen for ASCA, which is actually a glycan. And these ASCA IgGs were isolated from the immunized mice, inoculated in a recipient mice, followed by the induction of the colitis. And actually, we demonstrated that mice inoculated with ASCA displayed an increased disease severity, as you can see here by this disease activity index. And in this case, we also observed an activation of the dendritic cells. But then we had, as usual, a tough question from a reviewer asking that we needed in this model to prove the Fc gamma receptor interaction, to prove that ASCA is really pathogenic. So we needed to ask, I would say, to the world about a quadruple Fc gamma receptor knockout, and we managed to have from Harvard lab and Mark Crabb from Southampton. And what we did was exactly the same, but in this way in the Fc gamma receptor knockout mice. As you can see here, in the Fc gamma receptor knockout mice inoculated with ASCA, there is a significant decrease in the susceptibility to develop colitis, alongside a decrease in the pro-inflammatory environment, as you can see by the decrease in IL-1 beta. So taken together, we propose that glycan can actually trigger a novel immunological program mediated by glycans or changes in glycome that can activate or modulate immune response years before the diagnosis. But the relevance of glycans in the preclinical phase comes in other flavors. This was our contribution also to this study, in which neutralizing anti-GM-CSF autoantibodies that were also demonstrated to be upregulated in the preclinical phase. What we demonstrated is that these neutralizing anti-GM-CSF autoantibodies recognize for a specific glycoform of GM-CSF that can be detected years before diagnosis. So this is where we are in the glycan trigger. It's a consortium with 9 partners. One of them, we have the collaboration with Manfred at LUMC. But now I'm going to show you how we can actually propose glycome or changes in glycome as a common denominator of different immune-mediated diseases. And this is where I'm going to show you how glycan trigger and glycan switch project, in which we are gladly involved in, can be somehow interconnected. And I would like to tell you the example of these anti-glycan or anti-post-translational modification antibodies, and the example of ASCA in IBD, in Crohn's disease, and the example of ACPA in RA. So ACPA stands for the anti-citrulline peptide antibodies that can occur associated with rheumatoid arthritis. So as I told you, we have evidences that these ASCA anti-glycan antibodies can occur predominantly in Crohn's disease and can be detected in the preclinical phase. And the same holds true as a result of years of research by our collaborators and colleagues and partners in the glycan switch, Tom and René, that in fact high titers of ACPA can be found in the preclinical phase, so preceding rheumatoid arthritis onset. So bringing glycans to these, we know that we have in IgG glycans not only modulating the Fc portion, but also the Fab portion, meaning the antigen recognition domain of the immunoglobulin. And starting to the Fc, independently, the group from René Tos and Tom Huizinga demonstrated that, in fact, changes in ACPA Fc glycosylation can precede disease onset in the case of RA. And we also demonstrated the same holds true for ASCA, preceding the onset of Crohn's disease. So related with the Fab glycosylation, we have the seminal contribution of the René and Tom's group demonstrating that this V domain glycosylation occurs in ACPA and can precede disease onset. What we are trying to do in the glycan trigger project is to see whether ASCA Fab glycosylation is also altered. And this is Leticia Gomes, which is a postdoc hired in the glycan trigger project. And these are some preliminary evidences, it's not yet published, but we are chasing on these, that in fact at least there is some mass difference between the ASCA and our controls that may support the fact that ASCA Fab glycosylation can be a contributor for the sake of the Crohn's disease. And what we are in this interconnection between the glycan trigger and glycan switch is to tackle this from the B cell level. So it's the main cells responsible for antibodies production. So what happens at the B cell level? And this is the talk of Ines Alves of tomorrow. So I'm finishing by thanking my amazing group, to all of the collaborators, some of them here represented. We do a truly translational research, and I'm happy to take any question from you. Thank you.
It was a really phenomenal talk. So is there a link between the change you see on the T cells in Crohn's with the antibody responses?
That's a very pertinent question.
Yeah.
And we do know that B and T cells can somehow interact at the level of the germinal center. This is something that we are tackling in the glycan switch at the level for RA, but it's a very relevant question also for the Crohn's disease. Yeah.
And then, second question, sorry. Is the change you see in the T cell, is that a global thing or is it antigen specific? The reason I ask is that there's actually Nico is doing this. A lot of people doing it where they're modulating CAR T cells and T cells to reduce MGAT5. Is that going to be a bad thing if they do it?
Yes. Actually, we're tackling this in another project, which is in colorectal cancer. We are focusing also on the T cells glycosylation, taking into consideration these evidences that the type of glycans in T cells define the effector functions of the T cells, which is also very relevant for anti-tumor effect, and also because we demonstrated that the way that the T cell is glycosylated can be defined very early in thymic T cell development. So that's very pertinent. Yes. Thank you.
So very nice talk. Thank you very much. So I have a question on the ASCA antibodies, which I assume, and I think I saw it also in your data, are mostly IgG2 antibodies, right?
Yes.
And I also assume, so that makes it a lot of sense that you only look at Fc gamma receptor 2. But I was just also wondering, so from the glycosylation point of view, you only measure total IgG, or did you also manage to measure antigen specific?
Yes, initially, this was something that was performed by Gordon. We measured total IgG glycome. But then at the functional level, because we captured the ASCA IgG, we did some galactosidase tests to see whether this is specific for ASCA. And what we are doing now with Manfred in the glycan trigger is to purify ASCA specifically and to characterize IgG. IgA.
Very nice. So that's coming. So, and also just to follow up on this, my first question. So, predominantly IgG2 does not mean that it's not no IgG1 or no IgG3. Do you know if there is also some IgG1 and or 3, and to what level?
Yes, we analyze it at the subtype specific level, IgG1, 2, 3, and 4.
No, so I mean, is it also present? Do you know if it's present and to which level?
Then we decided to focus on the IgG2, H3, and 4, F1 because of this strong connection with ASCA. But other changes in glycosylation was also observed in other
And I mean
subtypes.
also for your functional testing with dendritic cells, I mean, if there is a part of them, of the antibodies, like 10% being IgG1, you'll also get contribution from Fc gamma receptor 1 and possibly 3 if it's present.
Yeah, that is something that we will need to figure out, what the proportion of contributions of the different subtypes. Yeah. Thank you.
Tom has a question.
Thank you.